Hello everyone. Today we will discuss about membrane structure, dynamics and application. As we know that the membrane composed of lipids and protein from the barrier between each cell and its environment. Membranes also partition the cytoplasm of eukaryote into components including the nucleus and the membrane-bound organelles. Each type of membrane is specialized for its various functions, but all the biological membranes have much more in common that are a planar fluid bilayer of lipid molecules, integral membrane protein that crosses the lipid bilayer, and peripheral membrane protein on both surfaces. This video opened with a discussion of lipid bilayer, then considered example of integral and peripheral membrane proteins. Before concluding with discussion of the dynamics of both lipids and protein, we will discuss about some application of the plasma membrane. So myself, Dr. Deepak Verma. First we discuss about the development of ideas about the structure. So our current understanding of membrane structure began with very early in 1895 that a cellular membrane consists of lipid bilayer. In 1920 it was found that the lipid extracted from the plasma membranes of red blood cells spread out in a monolayer on the surface of the tray of water to cover an area sufficient to surround the cell twice. Yes, as you see, first when 1920s around, there is no any protein. These all are lipid bilayer. There is no protein. Yes. During 1930s, cell physiology realized that a simple lipid bilayer could not explain the mechanical properties of plasma membrane. So they postulated a surface containing of protein to reinforce the bilayer. Yes. See, in 1943, some proteins are lies on the surface of the plasma membrane. After that, in early electron micrograph of thin sections of the cell straightened this view, since all the membranes appeared as a pair of dark lines separated by a lucent area. But in 1970, now we come to the 1970s, two complementary approaches showed that protein crosses the lipid bilayer. First, electron micrograph of membranes that are split into two while frozen. This technique is known as nowadays as freeze fracture technique. Revealed protein particles embedded in the lipid bilayer. Later, chemically labeled showed that many membrane proteins are exposed on both surfaces of bilayer. Light microscopy with fluorescent tags demonstrated that membrane lipid and some membrane proteins diffused in the plane of the membrane. Quantitative spectroscopic studies showed that lateral diffusion of lipid is rapid, but the clipping from one side of a bilayer is other is slow. This is fluid mosaic model of membrane. Yes, this this model. This one. This one is fluid mosaic model. See, these are the integral proteins. These are the intrinsic proteins. Crossed by layer. Here, these are the proteins. Okay, here also. Now, the fluid mosaic model of membrane is incorporated this information showing transmembrane protein floating in a fluid lipid bilayer. Subsequent work revealed structure of many proteins that span the lipid bilayer, the existence of lipid anchor on some membrane proteins, and network of the cytoplasmic protein that restrict the motion of many integral proteins. Nowadays, we will here, and these are the ultra model of fluid mosaic model. Then density of protein is actually membrane is higher than the electron, as you seen in the figure. This picture showing the actual fluid mosaic model for biomembrane. Here we can see these are lipid bilayer molecules, and this side is cytosol and this is the cell exterior. So you can see peripheral membrane proteins are lying on the surface of there, and these are the integral proteins. This one, this one, this one, this one, this. These are the integral protein, integral membrane protein. And this is also peripheral membrane protein, which is anchored with the cytoskeleton here. Now, one thing I want to share with you that fluid mosaic model given by the Singer and Nicholson. Now we know that the membranes are composed of lipid and protein. So first we discuss about the lipid bilayer. So membrane lipids are amphipathic molecules. So what is amphipathic? Amphipathic means that molecules that have polar and non-polar both properties. So, and lipid or fatty molecules constitute about 50% of the mass of most animal cell membrane. Nearly all the remainder BTE protein. There are approximately 5 billion lipid molecules in one square micrometer area of lipid bilayer. All the lipid molecules in cell membranes are amphipathic. All, yes. Means they have hydrophilic, water-loving or polar and hydrophobic, water-fearing or non-polar. The most abundant membrane protein lipids are phospholipids. These have polar head group of and two hydrophobic hydrocarbon tails. The structure we will discuss later. And another class of membranes are glycolipids that have some carbohydrate part and sterol. They are just like cholesterol. Yeah. Here I want to also share some important thing that cholesterol or sterol mostly found in animal cell membrane. They never found in plant cell membrane. And glycolipids, they are only present on plasma membrane, never the cell organelles membrane like ribosome. Ribosomes never have a membrane, mitochondrion, nucleus, all the things. That's why some glycolipids act as the antigen. This is the basic structure of phospholipid. Here you can see a head and tail. So this is hydrophobic head, and this is from here to here, this is hydrophobic tail. So this is polar and this is non-polar. This is always membrane exterior, this is interior side, and this is for exterior side. So phospholipid made up of mainly 4 components. First, an alcohol. In most of the cases, this is glycerol, and sometimes it's sphygmocin, and fatty acids, 2 fatty acid chains, 1 phosphate, and 1 another alcohol. Most of the time, this is a choline. 4 major phospholipids predominate in the plasma membrane of many mammalian cells. Everywhere you can see that this is the common feature here. So this is glycerol, but this is not the glycerol molecule. This is actually a cyclosin molecule, and see, this is phosphate, this is phosphate, 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 and then phosphate. And another thing is the alcohol. See, this alcohol, this is the alcohol, this is also a part of alcohol, this is also alcohol. And these are the fatty acid tails here, 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 and here. So these all four phospholipids are found in plasma membrane. Together, these phospholipids constitute more than half of mass of the lipid in most of the membrane. Now we come to the glycolipid and sterol molecules. Glycolipid contain carbohydrate covalently attached to the lipid. These can derive from either glycerol or spironolactone. The simplest glycolipid class, called cerebroside, contains a single sugar residue such as glucose and galactose. The complex glycolipid called globoside contains oligosaccharides. Gangliosides are globosides containing an oligosaccharide with one or more salic acids linked on the sugar chain. The glycolipids are found exclusively in the outer leaflet of the plasma membrane. And now to the sterol. The basic structure of sterol is a steroid nucleus. Okay. Now again, okay, this is the nucleus consisting of four fused rings, 1, 2, 3, 4. Four fused rings, 3 with 6 carbon and 1 with the 5. This is planar and relatively a rigid structure. Cholesterol is the major sterol present in the plasma membrane of all animal cells. The plasma membrane of plant cells lack cholesterol, but they contain other sterols like stigmasterol, with the exception of like mycoplasma. Bacterial plasma membrane also lacks the cholesterol. Up to here we had seen the structure of lipids in membrane. Now we comes to the dynamics of the lipid. The shape and amphiphilic nature of the phospholipid molecules cause them to form bilayer spontaneously in the aqueous environment. Hydrophobic molecules, by contrast, are insoluble in water because of almost all of their atoms are uncharged and nonpolar, and therefore cannot be energetically favored in interaction with the water molecules. If dispersed in water, they force the adjacent water molecule to recognize into ice-like cages that surrounded by hydrophobic molecules. See, you can see it here in my picture. This is polar heads and these are the non-polar tails. So this structure is known as mycelium. Because of these cage structures are more ordered in the surrounded by water, the information increased by free energy. This free energy cost is minimized. However, the hydrophobic molecules or hydrophobic portion of the amphipathic molecules clustered together so that the smallest number of water molecules is affected. When these amphiphilic molecules are exposed to an aqueous environment, they behave as you would expect from the above discussion. They spontaneously aggregate to blur their hydrophobic tails in their interior, where they are shielded from the water, and they expose their hydrophobic head to the water. Depending on their shape, they can do this in either two ways. They can form a spherical mycelium, like here, mycelium, and with the tail inwards, or they can form double layered sheets or bilayer. See, double layer sheet. One layer is here and one layer is here, with the hydrophobic tail sandwiches between the hydrophilic head groups. See, this heads are here also, here also, in between all these are the tails. Around 1970s, researchers first recognized their indivisible lipid molecules are able to diffuse freely within the plane of lipid bilayer. The initial demonstration came from the studies of synthetic lipid bilayer, which can be made in the form of spherical vessels called lysosome, as we seen earlier, or in the form of planar bilayer found across hole in the partition between two aqueous compartments on a solid surface. Now, various techniques have been used to measure the motion of individual lipid molecules and their components. One can construct the lipid molecule, for example, with the fluorescent dye or a small gold particle attached to its polar head group and follow the diffusion of each individual molecule in the membrane. Alternatively, you can modify a lipid group to carry a spin level such as nitroxide group. This contains an unpaired electron whose spin creates a paramagnetic signal that can be detected by the electron spin resonance ESR spectroscopy, the principle of which is the motion of and orientation of a spin-level lipid in a bilayer can be deduced by the ESR spectra. Such studies show the phospholipid molecules in synthetic bilayer very rarely migrate from monolayer on one side to the another side. This process is known as flip-flop, occurs on a time scale of hours for any individual molecules. Although cholesterol is an exception to this rule and can flip-flop rapidly. In contrast, lipid molecules rapidly exchange places with their neighbors within a monolayer. This gives rise to a rapid lateral diffusion. With the diffusion, laterally diffused, they are each. So this type of mobilization in the phospholipid molecules. This one goes to here, this goes to here, or this goes to here. This is the lateral diffusion, and this is very fast. But this flip-flop is very rare. Rotation is also there. Rotation, and relaxation also. Despite their fluidity, lipid bilayer can form domains to different compositions. Because of a lipid bilayer is a two-dimensional fluid, we might expect most type of lipid molecules in it to be very well mixed and randomly distributed in their own monolayer. The van der Waals attraction forces between neighboring hydrocarbon tails are not selective enough to hold groups of phospholipid molecules together with certain lipid mixtures in artificial bilayer. However, one can observe phase segregation in which specific lipid comes together in separate domains. There has been a long debate actually among the cell biologists about whether the lipid molecule in the plasma membrane of a living cell similarly segregate into separate domain. This is called lipid raft. Although many lipids and membrane proteins are not distributed uniformly, large-scale lipid phase segregation are rarely seen in the living cell membrane. Instead, specific membrane proteins and lipids are seen to concentrate in more temporally dynamic fashion facilitated by the protein interaction. They allow the transition formation of specialized membrane region. Such clusters can be tiny nanoclusters on a scale of few molecules or large assembly that can be seen with electron microscopy, such as cavilli involved in the endocytosis. Here, actually, here is I have seen a model for the raft domain. Here you can see weak protein lipid and lipid interaction reinforce one another to partition to interacting components in the lipid raft domain. Cholesterol, mm, cholesterol, sphingolipid, glycolipids, glycosyl phosphatidylcholine inositol, GPI anchor proteins in short form, and some transmembrane proteins are in these domains. Note that because of their composition, rhabdomins have an increased membrane thickness. We discussed glycolipid, GPI anchor protein, and oligosaccharides. Later somewhere. Now we move to the membrane proteins, another part of membrane. Although the lipid bilayer provides the basic structure of biological membranes, the membrane proteins perform most of the membrane's specific tasks and therefore give each type of cell membrane its characteristic functional properties. Accordingly, the amount that types of protein in membranes are highly variable. Such as example, in a myelin membrane, which serve mainly as electrical insulation for nerve axons, less than 25% of membrane mass is protein. But in contrast, in membranes involving ATP production, such as internal membrane of mitochondria and chloroplast, approximately 75% is protein. A typical plasma membrane is somewhere in between, with protein accounting for about half of its mass. Because lipid molecules are small compared to the protein molecule, however, there are always many more lipid molecules than in protein molecules in cell membranes. About 50 lipid molecules for each protein molecule. In cell membrane, there are 50% protein by mass. Membrane proteins vary widely in structure and in the way they associated with lipid bilayer, they reflect their diverse functions. Here we can see proteins are associated with bilayer in various ways. There are 8 ways up to the. This picture shows the different ways in which proteins can associate with the membrane. Like their lipid neighbors, membrane proteins are amphiphilic, having hydrophobic and hydrophilic regions. Many membrane proteins extend through the lipid bilayer and hence called the transmembrane proteins, with the part of their mass on either side. I am showing here. This is transmembrane protein. This one. See, these are here is lipid is inbuilt with the lipid bilayer. Their hydrophobic regions. See, they are hydrophobic. This is hydrophobic region. This is hydrophobic regions. Their hydrophobic regions pass through the membrane and interact with the hydrophobic tails of the lipid molecules in the interior of the bilayer, where they are sequestered away from the water. Their hydrophilic regions are exposed to the water on either side of the membrane. This hydrophilic part. This is hydrophilic. This is hydrophilic. This is hydrophilic part. This is hydrophilic part. Exposed to water. The covalent attachment of fatty acid chains that insert into the cytosolic monolayer of the lipid bilayer increases with the hydrophobicity of the sum of these transmembrane proteins. Other membrane proteins are located entirely in the cytosol and are attached to the cytosolic monolayer of lipid bilayer either by amphiphilic alpha helix exposed to the surface of the protein, just like in this example number 4. This is the cytosolic part, and this is exterior. So this is cytosolic part, this attached with this one, or by one or more covalently attached to the lipid chain. As you see in here, this is attached with the lipid molecules. See, this example number 5. Yet other membrane proteins are entirely exposed at the external cell surface, being attached to the lipid bilayer only by the covalent linkage via specific oligosaccharide to lipid anchored in outer monolayer of the plasma membrane. Yes. So these are the lipid molecules, this carbohydrate, these are the carbohydrate chain, this phosphate, and this is liquid, this one. Yes, example number 6. Okay, this is outer side. This is never occurs in here, this side, because this is carbohydrate part, and sometimes this act as an antigen. This antigen. The lipid linked protein in example number 5 are made as soluble protein in the cytosol and are subsequently anchored to the membrane by the covalent attachment of the lipid group. The protein in example number 6 however are made as a single pass membrane protein in endoplasmic reticulum. While still in ER, the transmembrane segment of the protein is cleaved off and a GPI anchor is added, leaving the protein bound to the to the non-cytosolic surface of the ER membrane solely by the anchor. Transport vesicles eventually deliver a protein to the plasma membrane. Now we comes to the example number 7 and 8. By contrast of these examples, membrane-associated proteins do not extend into the hydrophobic interior of the lipid bilayer at all. They are intended bound to the either surface of the membrane by non-covalent interaction with other membrane proteins, as here 7 and 8. Many of the proteins of this type can be released from membrane by relatively gentle extraction procedures, such as exposure to the solution is very high or low ionic strength of extreme pH, which interfere with protein-protein interaction but leave the lipid bilayer intact. These proteins are often referred as peripheral membrane proteins. Okay? These are actual peripheral membrane proteins. Transmembrane proteins and many proteins held by bilayer by lipid groups, hydrophobic polypeptide region, and insert into the hydrophobic core of the lipid bilayer cannot be released by these way. Now we move to our last part, that's the membrane function. So just an overview to the membrane functions. We go by step. Number 1 is the compartmentation. So membranes are continuous unbroken sheet and as such inevitably enclosed compartment. The plasma membrane encloses the contents of entire cell, whereas the nuclear and cytoplasmic membranes enclose diverse intracellular space. The various membrane-bound compartments of cell process markedly different contents. Membrane compartmentalization allows specialized activities to proceed without external interference and enable cellular activities to be regulated independently by one another. Next is the biochemical activities. Scaffold for biochemical activities. Membranes not only enclose compartment but also act as a distinct compartment themselves. For reactants floating around in solution, the interaction are dependent on random collision. In contrast, components that are embedded in the membranes are no longer floating free and can be ordered for effective interactions. The next is the security, just like a selectively permeable barrier. Membranes prevent the unrestricted exchange of molecules from one side to the other. At the same time, membranes provides the means of communication between the compartments they separate. The plasma membrane, which encircles a cell, can be compared to the moat around a castle. Both serve as a general barrier, yet both have gated bridges that promote the movement of selected element into and out of the enclosed living spaces. Next is very important, transportation. The plasma membrane contains the machinery for physically transporting substances from one side to the another, often from the region where the solute is present at low concentration into a region where the solute is present at much higher concentration. The membrane transport machinery allows a cell to accumulate substances such as sugars, amino acids, and are necessary to fuel its metabolism and build its macromolecules. The plasma membrane is also able to transport specific ions, thereby establishing ionic gradients across itself. This capacity is especially critical for nerve and muscle cells, and in detail we will discuss in this phenomena in detail. Next is the response to the external stimuli. The plasma membrane plays a critical role in the response to the cell to external stimulus process known as signal transduction. Membrane possess receptors that combine with specific molecules known as ligands or responds to the other type of stimuli such as light or mechanical tension. Different types of cells have membranes with different receptors and therefore capable of recognizing the, and responding to the different environmental stimuli. The interaction of plasma membrane receptors with an external stimulus may cause the membrane to generate signal that stimulate or inhibit internal activity. For example, signal generated at a plasma membrane may tell a cell to manufacture more glycogen to prepare for cell division or to move forward a higher concentration of a particular compound to release calcium from internal stores or possibly to commit suicide as apoptosis. Uh, Next is the intracellular interactions. Situated at the outer edge of the every living cell's plasma membrane of multicellular organism mediates the interactions between the cell and its neighbor. The plasma membrane allows cells to recognize the signals one another to adhere when appropriate and exchange material and information. Proteins within the plasma membrane may also facilitate the interaction between extracellular material and the intracellular cytoskeleton. I think the last is the energy transduction. Membranes are immediately involved in the process by which one type of energy is converted to another type. That is known as energy transduction. The most fundamental energy transduction occurs during photosynthesis when energy in sunlight is absorbed by membrane-bound pigments, converted into chemical energy and stored in carbohydrates. Membranes are also involved in the transfer of chemical energy from carbohydrates and fat to the ATP. In eukaryotes, the machinery of these energy conversions is contained within membranes of chloroplast and mitochondria. Yeah, that's it. Thanks for listening.